right. Hello, everyone. My name is Anna, and my research project this semester. Can you all see my screen? Yes. Okay.
Put it on slideshow mode, Anna.
Yes, of course. I just remembered. All right. All right. Hello, everyone. My name is Anna, and my research project this semester is about using bioinformatics as a tool to create a plant DNA barcode. I did this under the guidance of Professor Nalini Odapalli here at Valencia College. DNA barcoding is a method in bioinformatics that helps us to classify the species from the DNA by taking a characteristic portion of a gene known as RBCL. The characteristic portion of the sequence helps us to generate a barcode to compare the sample to every species. The RBCL gene is located in the plant chloroplast, and it is highly conserved, meaning that it is very similar across species. This process, according to President Erickson in 2012, requires a library of known DNA sequences to compare against, which bioinformaticians have been doing for the past 20+ years. The software I used for this project, DNA Subway, does all of this for us by taking our sequence and comparing it to a library of known sequences from thousands of other species. It then takes this comparison and puts it on paper by generating a phylogenetic tree, which shows the evolutionary similarities and differences between these species. In essence, the question we're asking is, what species is the unknown sample's DNA sequence most closely related to? My null hypothesis is that the unknown sample might not be related to any plant species in the database, and my alternative hypothesis is that it is related to one of the plant species in the database. To understand how all of this works, we have to take a deeper look at DNA barcoding itself. In 2003, Heppard and his colleagues produced a research study which defined DNA barcoding as a technique. Although DNA barcoding has been around since 1994, this article standardized the method we use today. Using this method, they were able to create a giant phylogenetic tree, which pictures the relationship between many different species. This showed scientists that creating a DNA barcode is a great way to identify species. Thanks to Heppard and his colleagues, anyone with a computer can now identify almost any organism on this planet. As I mentioned before, I used a bioinformatics tool called DNA Subway. First, I created an account as a Valencia College student and a project, as you can see at the bottom. And I used their glue line process for this project. I was given an unknown sequence from a previous undergraduate researcher under the guidance of Professor Nalini. This sequence was obtained from a sample randomly picked from a field. I entered the unknown sample sequence into the corresponding field, and then I noticed that there were many gaps between the forward and reverse sequence. There were also overhangs, as you can see right here at the beginning, which are at the end of either forward or reverse sequence, and they do not have a corresponding match. As you can see in the top left of this picture, I trimmed the sequence to reduce the overhangs and thereby increased the sequence alignment. I also matched the forward and reverse pair of sequences. All of this research was done via Zoom. As you can see here, this is one of our meetings. And after that, I used BLAST, which stands for Basic Local Alignment Search Tool, which is a program that compares the sequence identity, a similarity between our sample and other DNA sequences within the database. The next step is that I had to select species in the plant kingdom for this phylogenetic comparison. And there were many plant species and categories that I could have chosen from. For example, monocots, dicots, angiosperm, and gymnosperm. Here you can see my phylogenetic tree that was made by DNA subway. The base of the tree shows the common ancestor, while the branches show where each organism splits from one another. The tree shows us that my unknown sample was most closely related to the snapdragon Plantagenaceae family, meaning that it is most likely a snapdragon. We can also see the similarity between my sample and other related plants, such as sunflower. Brachytaceae in red is an outgroup or species which directly descended from a common ancestor, and it is not very closely related to the sample. This helps to route the rest of the members of the tree to their relatives. The numbers at each branch show the level of confidence in the placement by how many times DNA subway modeled this relationship with the data given. Generally, anything above an 85 is seen as fairly reliable, but due to the small sample size and a few missing known sequences, some of these numbers are less than ideal. However, for our purpose of the direct comparison, they're good enough. This is my DNA barcode, and you can see the different colors that show the different nucleotides. And DNA subway generated this through bioinformatics and the application which I learned to use during this research course. Because the sequence is highly conserved, we can assume that the sample has similar properties as the plants related to it. The two most similar plants are snapdragon and sunflower. Snapdragon, according to Ramadan and El-Shami in 2012, has high linoleic and oleic acid content, which make it very nutritionally valuable. It has also been used for its high anthocyanin concentration, which is an antioxidant with a deep red pigment, giving the plant its vivid color. In 2008, Odapalli and colleagues used the gene for anthocyanin to make high nutrient tomatoes through gene transplantation. Here in this picture, you can see the difference between the tomatoes. The purple one is the one with the anthocyanin gene, and the other one is a normal tomato. Other studies, such as Adeleke and Babalola's 2020 article, state that sunflowers have high nutritional and pharmaceutical value, which Gogay and Jom confirmed in 2017, stating that sunflower seeds are high in oil and linoleic acid, just like snapdragon. There is a high chance that the genetic similarity between these three plants could mean that my sample is also nutritionally valuable, which could be concluded by doing more research. To summarize all of this, the most closely related species to my sample is snapdragon, a highly nutritional family of flowers, which implies that our sample could be useful for similar purposes. Further research has to be conducted on the sample to see if these characteristics are actually conserved, or if they're lost in the small differences in the genetic sequence. Therefore, I accept my alternative hypothesis that my sample will match with one of the sequences in the data bank, and I can reject my null hypothesis. And the unknown sample's DNA sequence is most closely related to snapdragon. Here are my references. And I want to thank Valencia College and late Dr. Brenda Schumpert for getting the fund here to do research at Valencia. And I especially want to thank my mentor, Professor Nalini Odapalli, for giving me this opportunity. And I also want to thank you for giving me the chance to present in front of you. And I'm happy to hear any feedback and answer any questions that you have for me. Thank you.
It was fantastic. Excellent. You said everything that I boosted into you, I stuffed into you. Wonderful. Thank you. Destiny, you want to go next? And then we will take the questions after her presentation.